Srivastava from CSIR CEERI Pilani for to deliver this session. I would like to introduce today's our expert, Mr. Dr. Satyam Srivastava. Dr. Satyam has completed his BTech from the University Institute of Engineering and Technology, Kanpur. Then he completed MTech in Advanced Electronic Systems uh, through a research work done at CSIR CEERI Pilani, and the degree was awarded through ACR University, New Delhi. Recently, he's completed his PhD through CSIR CEERI Pilani, and his degree awarded through ACSIR University, New Delhi. Satyam Srivastava is presently working as a scientist at CSIR Central Electronics Engineering Research Institute, Pilani. During his PhD, he worked in the area of non-destructive sensing techniques for fruit quality assessment, multimodal data processing, and data fusion algorithms at CSIR CEERI Pilani. His research area includes low-power embedded system design, multimodal data processing, and data fusion for water, food, and health monitoring application. He is currently associated in various projects of ultra-portable, ultra-low power systems on site measurement of water quality, wearable devices to measure vital health parameters of elderly disabled persons, and handheld IoT-enabled smart probe for cervical cancer screening. He has done numerous projects on agriculture, data processing, and field devices. So hope his session will help the participant to know more about the data processing for the fruit quality measure. So I request Dr. Satyam Srivastava to start with his presentation. Sir, please.
Thank you, Praveen. Thank you for this awesome introduction. So today we'll be discussing about different issues in the agriculture for fruit harvesting during the fruit harvesting time. So there are major two issues. One is pre-harvesting and one is post-harvesting. So in my presentation, I'll be discussing mainly in the pre-harvesting issues. So if we discuss about what are the food spoilage, basically what are the reasons behind food spoilage. So there are few of the facts with me. You can see in the presentation. So more than 25% of total food spoilage occurs due to on-field judgments like handling and storage. So what are the on-field judgments? Like our Indian fruit growers are not able to judge the optimal harvesting time. So when to pluck a fruit, if you will not able to predict it quite right, so it then this fruit growers need to use different artificial ripening methods, and which also affects the fruit quality, nutrition parameter, and also human health. And somewhere it causes to fruit, let us say, spoilage. So, so we talk about the data. So you can see that 83 million of metric ton fruits wastage every year, and due to wrong on-field judgments, and others are also like for handling and storage, and for this packaging and processing, distribution and consumption. So what are the different reasons behind this spoilage? So one is this premature harvesting, that if a fruit is not ready to harvest, it's still this different fruit growers harvest it and use different artificial ripening methods like chemicals and these kind of things to ripen, to apply force ripening methods. Now apart from that, this shelf life judgment. So after harvesting it, how much time a fruit grower can hold into their cold storage, or let us say how much time they can hold overall fruit samples on the shelf. That is called shelf life judgment. It is also very much important parameter for a fruit grower to predict it. Then obviously various hazards, disease attacks are there with the fruit growers. So how they can actually recognize it, that this is the disease, and for every disease there are different kind of disinfection process is there. So this is one of the important things. Then this various adulteration practices. You all of you, most of you are aware that with different kind of this fruits, people have to use different injectables and to increase the weight, to increase the shelf life of the fruit. So these kind of practices are also there. Then improper storage houses. Obviously, then in Indian scenario, this storage, cold storage and storage also are not that much decent, so that this also affects the fruit quality. So there's another data. If we talk about the fruit only, so this was the Indian data, and this is the data I have collected all around the world. So how much money is involved in the fruit wastage? So we talk about it's around 142.5 US billion dollar money involved every year, waste globally due to fruit spoilage. So you can see it's a very serious matter. It's a very huge chunk of money which is involved due to this fruits spoilage. So we talk about majorly I'll be discussing about on-field problems because this is one of the big area, and this is something Indian fruit growers are struggling to solve these kind of issues. So to solve these issues, myself and my team have worked from, let us say, this past 5 years, and developed some very handheld and smart solutions to solve all these problems. So that I'll be discussing later time of the presentation. So we talk about what are the on-field problems. Obviously, I'll discuss already about this harvest time judgment, shelf life judgment, and this early warning of the disease attack, appropriate solution of infection. So if there's an infection is there in the fruit, then what is the solution for that? Then overall fruit quality index, whether it is let us say ready to harvest or not ready to harvest. If fruit is not ready to harvest, after how much time it will be ready to harvest. Then overall yield prediction. Obviously, a fruit grower should have an idea that in his orchard, what is the in his orchard, how much percentage of the yield is ready to harvest, how much percentage of yield is still not ripen or under ripen kind of a scenario. We talk about some of the facts. So optimum maturity of the harvest is a very important determinant to the fruit, final quality of the fruit. Obviously, if you will pluck any fruit at its optimal maturity, it will be having best nutritional parameters as well as best in the quality. And then we talk about a fruit picked too early and too late. So if you will pick unripened fruits, or let us say over ripened fruits, so they are susceptible towards different physiological disorders, and also have shorter shelf life. Shorter shelf life, that you cannot hold them after plucking for a long time. You need to use different chemicals and different artificial methods to hold them for a long time. Then obviously this I have already mentioned that fruit picked immature has been processed through various hazardous artificial ripening methods using different hazardous chemicals. Maturity index so obviously if you want to predict the maturity index, we need to actually measure the different parameter which is the function of maturity index. So like shape, size, color, weight, aroma, there are so many parameters, and it's very complex to judge a quality of a on-tree fruit. So we talk about what are the sensing methods available in the literature, and different researchers have contributed in this area. So we talk about what happened initially. So initially this manual inspection and chemical methods has been used by different researchers as well as in different fruit factories, fruit product factories. So generally people have used this manual inspection method using skilled manpower. So they generally smell it, they generally press it, and based on that they suggest that this is a good quality of the fruit, this is the bad quality of fruit. But in this, a lot of human intervention is involved, and there is a fair probability to fail the test, and there's a fair probability of that error can happen. So already most of you are aware that this few years back there was a very big issue regarding this alfonso. India have transferred this alfonso whole chunk got rejected from US because some of the lot got spoiled. So based on that, they have rejected overall slot of our alfonso mango. So we talk about there are several chemical methods also earlier people have used. One of the famous method is iodine spray test. So what they have done, they they select some of the samples out of the chunk and they cut it and they spray the iodine, and based on that change in the color of the fruit sample, they judge that whether fruit is ripen or under ripen. Generally, this kind of test happens with apple samples. After that, this few of the researchers have introduced this mechanical methods like penetrometer, instrumented hammer, impact. So in these kind of instruments, they generally apply pressure on the fruit sample using these mechanical instruments, and based on that, they judges the overall stiffness and firmness of the fruit sample, and based on that, they actually, let us say, correlate with the ripeness and harvesting time of the fruit sample. But these kind of methods have problems that if once you have used this method on any kind of fruit sample, then it will not be able to, let us say, consume by human, and it will total damage. And other issues that these kind of methods you cannot use on field. So these kind of method you can use on some kind of fruit sample which you have plugged and which you are, let us say, in this lab or in cold storage. Then few of the researchers have this shared this destructive type electronic methods like impedance measurement technique, GCMS based technique. So these methods are also lab based methods, and these instruments are very much heavy and bulkier. So in this GCMS is very famous technique. So in this they generally used to extract the tissue of the fruit sample and then go through this standard technique, and based on that they extract the different VOCs and different chlorophyll and different ripening compounds of the fruit sample, and based on that concentration of different compounds, they predict that this lot is ready to harvest, or these kind of things. Then this non-destructive methods, contact type, like capacitive based measurement. So in this measurement, generally this capacitive type sensors has been developed by different researchers, and then they have actually measured the capacitance of different fruit samples, and based on that they try to correlate with the harvesting time and different quality parameters of the fruit. Then this is the recent one, which is the non-destructive method, which can be used in the field also. So our aim was to how we can develop hand-held gadgets which can be useful for different fruit growers, and which they can use in their daily life to judge the optimal harvesting time or different quality parameters of a on-tree fruit, so that they, based on that, they can decide that this chunk of their yield is ready to harvest, or they can take a right decision. So basically in our whole this work, we have explored 4 different sensing techniques. First one is electronic nose. So basically in this technique we have tried to mimic the human nose with the electronic nose, and to sense the different VOCs of the fruit sample, and based on that it can predict the overall quality of the fruit sample. Then the second is the machine vision. Machine vision is just like a mimic to a human eye. So how you can actually, using this technique, we can actually extract the outer surface of the fruit sample, color, shape, size, these kind of things. Then spectroscopy. Spectroscopy is also one of the important technique which we have used to judge. So apart from the smell and outer surface, there are different compounds which are very important to judge, like this chlorophyll, then total soluble solid. There are so many compounds which are very important to judge the overall quality of the fruit. So these compounds will tell you that what is the nutritional factor of a fruit. Then ultrasound technique we have used basically to judge the stiffness of the fruit. So just like human press a fruit, so we have developed this ultrasound based non-contact device which actually tells you the stiffness and firmness of the fruit sample, and based on that it predicts the overall fruit quality of the device, fruit quality of the on-tree fruit. So what are the global status? So if you talk about the Indian status, there's no such kind of device available in the India, or let us say manufactured by India. These are few of the devices which you are seeing on the screen, developed globally. And so if you talk about the first device, it is the F750 handheld bricks dry matter meter from Felix Instruments. It's a USA based company. So it just measures the sweetness of a fruit, and based on that sweetness it predicts that whether your apple or mango or whatever samples are ready for harvest or not. So generally they suggest that they use for this cherry, apple and Sirocco kind of fruit samples. And the second one is also so this is in this system they have used this spectroscopy, an IR based spectroscopy sensing technique to extract this total soluble solid, or let us say bricks compound in the fruit sample. Then second is this handheld non-destructive fruit tester from Korea. It is also uses this NIR based spectroscopy technique and uses almost similar kind of concept to measure this total soluble solid in bricks compound. Then this third is DA meter from Italy, and similarly this handheld. So this fifth one is from this USA. It is ultrasound based on ultrasound sensing technique, and it basically measures the let us say stiffness of the fruit sample. The last one is not for fruit. It is from this for this meat. It is based on electronic nose sensing technique, but it is not for fruit, so we do not discuss it. So if you talk about these systems are available globally, but you can see the cost of the this system. So each system is more than 5 lakh, 20 lakh, 8 lakh, 5 lakh. So these systems first, so there are few of the problems. One is this too much costly. Obviously, a Indian fruit grower cannot afford it. Second point is that it is only trained for other country environmental conditions and their crop conditions. So if we'll import these devices, these device has fair chances that they will not give good results to Indian environment as well as for Indian crop. And third point is obviously that it is specific for one particular this parameter, which is total soluble solid, or let us say bricks matter of the fruit sample. While as in our research and study, we have found that fruit quality depends on multiple parameters, not only one parameter. So by predicting a fruit quality based on one parameter is not very much good decision to tell the fruit grower. So we was talking about what are the things available in India. So you can see this is the status in India, which is available in which is fabricated. So these are lab-based devices, and these are totally destructive type devices. So these you can use to judge the stiffness of the fruit or chlorophyll of the fruit sample. But you need to break the fruit sample and you need to take out the juice of the fruit sample. So this is totally destructive type techniques, and you cannot use on field, or anyone fruit grower cannot use on field. So talk about what are the gaps exist in the literature and exist in the market. Based on that, our team have started doing research in this area. So I have already talked about that the whatever techniques are available in India, most are destructive type. Then whatever research available globally as well as in India, so they basically focused on single sensing techniques. So if we talk about these devices, what I have shown you right now, available globally, they only focused on single sensing technique, either spectroscopy, either electronic nose, either this ultrasound. So apart from that, algorithmic intelligence is one of the problem. So they does not introduce a very intelligent kind of algorithmic intelligence to predict different quality parameters, and then they also predict very limited quality parameters like PSS and obviously this accessibility and cost is one of the issue. Then Indian context, I already told you that these devices have been trained different environmental issues and different crop scenario. So which there's a fair probability that which cannot work according to Indian scenario. So in our research area, we basically focused on this non-destructive sensing techniques and how we can combine multiple sensing techniques to extract the multiple fruit quality parameters of a on-tree fruit. So we have used 4 different sensing techniques, and we have also tried to combine it and using different data fusion techniques along with different algorithmic intelligence to use different multimodal data processing techniques, and how we can actually extract most of the information from the fruit sample, and based on that we can judge that what is the overall quality of the fruit. So if we talk about the overall architecture, so in our research work, I already told you that we have focused on these 4 sensing techniques, and then we have developed this handheld hardwares to realize these sensing techniques, and this sensing method specific pre-processing technique, feature extraction technique, data fusion methods, and then correlation models have been implemented to predict these 6. So in our research journey, we have found that these 6 parameters are there, if we can judge it, let us say, or predict it accurately, we can actually judge the overall quality of the fruit. So one is the chlorophyll, second is obviously weight, third is volume, and fourth is total soluble solid or bricks, and third is this fifth is sweetness, and the last one is acidity. So then these quantitative parameters we have correlated with the qualitative decision, because our Indian fruit growers are not that much skilled that they can actually, by seeing these values of chlorophyll, weight, volume, they can just that this is a ripe and unripe. So we have converted these quantitative values into qualitative decisions. So like chlorophyll we have correlated with ripeness index of the fruit, weight and volume we have correlated with the maturity of the fruit, and these three parameters which are corresponds to taste of the fruit sample, we have correlated with the flavor of the fruit sample. So ultimately there are only three things: ripeness, maturity, and flavor of the fruit sample, which we can uh, we need to judge before harvesting a fruit sample, or let us say predicting quality of a overall fruit sample. So this is our first device. This is the handheld electronic nose. In this, we have used this metal oxide based gas sensors to judge the to extract the aroma coming out from different fruit samples. So we have developed different multiple iterations to achieve the final device, which is, you are saying, in the palm size device, the totally battery operated device. It is communicates with your smartphone based Android app and sends data to your third party cloud. So based on the cloud, you can run your algorithms and model, and then you can process that data from there. So there's no such kind of constraint that memory is less, and on field data processing you can need to done. So so this was which you are seeing in the top. On the top, this was our first kind of a model of a handheld electronic nose. But we have, after some time, we have found few of the issues like gas sensor array drift due to different environmental variations like change in temperature, change in humidity. So our results was quite changing with the different environmental issues. Then the purging cycle means there are two cycles in this electronic nose. One is aroma injection cycle, when actually our device takes aroma from fruit sample, and there's a purging cycle in which our device take out aroma from inside the device. So it is basically to for the cleaning the device, so that we can go for next testing of our next fruit sample. Then so in our earlier model, there was no purging cycle. Then this less sensitive towards target. So obviously the whatever VOCs we have targeted earlier, those are not very much appropriate. So in the latter model we have changed it. Then you can see this overall model was handled, but still it was bulky and consumes high power. Then obviously earlier we have trained with a very small data set, around 50 to 100 samples collected from nearby orchard. So whatever you are saying in the bottom, it was our final device. You can see it's a palm side device, quite low power, it's IoT enabled device, talks to your smartphone based app. So we have removed everything, touch screen and everything which was not required. So this is a little bit description about it. So this is the functional block diagram about. So device have a different gas sensors to sense the different VOCs coming out fruit samples, and these are the sensors information which we have used in the final model. And this is the odor injection and purging cycle scheme which we have implemented in our device. So this was the idea that first basically it device conducts the purging cycle to clean the sensing chamber, and then it conducts the odor injection cycle to take in the fruit aroma, and based on that it can store the response of different metal oxide sensors. So this is the critical response of our gas sensor array. So in our device right now we have used 6 gas sensors, and overall this response stores in our device and also on the cloud for further processing. So this is the prime. After developing it, we have tested it for different VOCs before just going for this fruit sample. So we have tested for different acetone, benzene, methanol to just to check the functionality of the device, whether the device is working fine or not. So in the real-time graph, it is not very much visible that our device is working or not. So we have implemented few of the classification techniques. So here you can see that our device is quietly working very fine, and it is able to classify between different BOCs. You can see acetone, benzene, and methanol, right? So these are few of the features. So in a critical gas sensor response, first we have applied this baseline correction to actually compensate the environmental issues and environmental drifts due to change in the humidity and temperature, which is changing on the field. Then we have extracted 5 different features like peak time, maximum peak time, settling time, fall time, and settle value. And these parameters, they are fed into our models to predict different fruit quality parameters. So first few slides of my presentation will talk about only about the hardware, and later few of the slides I'll be talking about the data coming from these devices and how we have processed it and how we have correlated with the actual fruit samples and how we have developed final model. So this was the first device. Now this is the second device, which I have already talked about, the ultrasound based sensing technique. So in this we have basically used 40 kilohertz ultrasound transmitter and receiver to transmit the ultrasound signal and to sense the reflected echo signal coming from fruit sample. And based on the received echo signal, we have analyzed it, and based on that we have tried to extract the different features of received signal, and based on that we have tried to correlate it to different fruit samples. So this is the handheld device, totally battery operated. Again, it talks to your smartphone based app. We have developed a custom app to communicate with it, and this app gives functionality to save the data to third-party cloud. So we have used this Microsoft Firebase cloud to store the data on the cloud, and further we can fetch from the cloud to process it. And this is the functional block diagram. So we've used this 8-bit microcontroller for this device, and this battery operated and battery monitoring, battery charging, and everything was there. So this was the specifications of the transmitter and receiver which you have used. So we have used this 120 degree beam angle for the transmitter and 40 kilohertz center frequency of the transmitter and receiver. So there are multiple modes in the literature which we can use to fix the transmitter and receiver. You can see there are 3 modes. One is this transmission mode, second is this pitch catch mode, and third is this pulse echo mode. So we have used this pitch catch mode to transmit the ultrasound signal and to receive the echo signal coming from the fruit sample. So obviously this stiffness varies at different part of the fruit sample. So we have thought how we can actually take out the spectrum of overall fruit. So we have divided a fruit sample into 4 different parts, and we have pointed 4 different points: data point 1, 2, 3, 4. 1, 2 on the vertical axis and 2 on the horizontal axis. So we have taken we have taken a spectrum on 4 different points for each fruit sample to generate the data set. And this is the internal architecture of the sensing chamber of this handheld ultrasound based device. So obviously between transmitter and receiver we need to fix on some angle. So we have conducted few of the experiments that on which angle we are receiving best response of the ultrasound signal. So we found that on 35 degree it is responding pretty much very well mannered. So we have fixed at 35 degree. So we have conducted some kind of preliminary testing before jumping for the fruits again. So we have taken randomly selected 3 fruit samples. Just a second. Yeah. So 3 fruit samples. One is this unripe, ripe, and third is overripe. And we have transmitted the ultrasound signal over them, and we have received the echo signal. So you can see the yellow one is the received signal, and blue one is the triggering pulse to the ultrasound transmitter. So you can see that A is our baseline when no fruit sample is there, and B is when we have placed unripe sample towards the sensing chamber, and this C is the ripe sample, and third is the overripe sample. So you can see as ripening is increasing, the stiffness of the fruit sample is also decreasing. So that's why it is absorbing overall almost all signal, and we are getting less amplitude into our reflected echo signal. So you can see the last D signal which is collected using our overripe sample, which is having amplitude is very less. So it tells that our device was working. Now we have thought how we can actually using, so this is the transmitted pulse which I was talking about, and this is the reflected echo signal. So we thought how we can actually extract the features. So basically in the ultrasound signal in literature also, there are two major parameters. One is the time of flight, how much time ultrasound signal taken from transmitter to receiver, and obviously the change in amplitude. So we have taken these two features to train our models. One is this obviously change in amplitude, which we have converted into attenuation, and second is the time of flight, which we have converted to propagation delay of that signal. So this is the architecture of data processing. So raw signal recorded from coming from surface of the CEERI sample. So we have selected CEERI samples for all our studies till now. So whatever you will see in my slides, you will see only CEERI fruit samples. Because there are nearby so many orchards, and we have collaborated with different ICR labs. So that's why we have selected primary citrus. While as these kind of studies, we are trying to generalize for other fruit samples also. So we have implemented this no frequency noise elimination because a lot of noise was coming into reflected echo signal. Then obviously time of flight and maximum amplitude extraction. Then conversion into attenuation and propagation delay. Then obviously these propagation delay and attenuation we have extracted on 4 different points of the fruit sample. So it is TPDX, TPDY, TPDZ, and TPW, and just like ATX, ATY, ATZ, ATW. Then we have fed into this statistical and artificial neural network models, which I will be talking later part of my presentation. And using this model, we have predicted these parameters. So now this is the third technique, which is spectroscopy sensing technique. So in this we have used different off-the-shelf existing light emitting diode based sources, and also different spectral engines to sense the radiation coming out of fruit samples. So this is the handheld device. It is having LED and spectral sensors to transmit the visible and NIR illumination, and to sense the reflected, or let us say reflected visible illumination and infrared illumination. Then we have designed this trigger control and UV-vis and IR's source readout circuit and micro-based communication circuit to your smartphone, which it can transfer data to your smartphone based app and further to the cloud. So this was the functional block diagram. So we have used this 6 LED based array, and along with this visible spectral sensor and this photodiode to actually transmit the illumination and to sense the illumination. So this is the details. So this is the spectral engine. We have spectral engine chip we have used, and these are the details of the LED. So we have used 3 different kind of LEDs: ultraviolet, UV from 340 nm to 420 nm, then white LEDs for visible illumination, then an IR LED for this 800 nm to 1130 nm. So this was the recorded spectral response by our developed handheld spectrometer. You can see it is for S1 fruit sample, and this is from 350 nm to 950 nm. And on the y-axis, it was a normalized distal reflectance, what we have got from fruit sample. So this is the custom enclosure design, because there are a lot of this angle and everything we need to take care in the mind for best response recorded. So we have designed our own custom enclosure and fabricated into our institute. So again, this for just going for testing of the fruit sample, we have thought to test whether our spectrometer is working for basic applications or not. So what we have done, we have actually taken 3 color cards. One is red, one is absolute red, one is absolute blue, and one is absolute this green. And we have recorded the reflectance using our own developed sample, our own developed handheld device. And we have found that it is pretty much change in the received reflectance. If you can see that on red one, it is a change reflectance on 650 nm, which corresponds to red color, while as in blue, it's a change reflectance in 500 nm, which corresponds to blue color. and on the green it is a change of 570 nm, which corresponds to green color. So these kind of experiments we have conducted so many times to verify the performance of the device and to whether our device was working perfectly or not. Then this is the data processing model. So in the raw spectral data recorded from our developed handheld system, we have converted into reflectance, and we have implemented few of this standard normal variate and this spectral noise elimination methods to remove the noise coming into the spectral spectrums. And this MSC based methods, multi-selective scattering correction, we have used to remove the background noise and background scattering coming into the fruit samples. Then this similar, just like in ultrasound, we have acquired a spectrum on four different points of the fruit sample. Similar strategy we have used for the spectroscopy also. We have acquired data on four points, and we have gathered four different spectrums: RS X, RSY, RXZ, and RSW. And these spectrums we have fed into our model for the training of our models, and also the average of all four spectrums we have also calculated and trained to our model. Then this is the fourth sensing technique, which is image processing based technique, and this machine vision based technique. So this in this we have used this developed this handheld device. This is our first model, which is very bulky. You can see it's a very bulky, and it consumes too much power, and it has different problems like results varies with the variation in the illumination conditions. So illumination independent problem was there. Whenever change in illumination occurs on the field, our results got corrupted. So what we have thought to actually implement few of the illumination independent algorithms, so how we can compensate the effect of illumination. Then obviously it is trained with a very limited data set. Then the camera, whatever we have used, that was very bad aperture, and resolution was not very much good. After conducting field trial, we have came up with these kind of these kind of points. Then this takes up very last time around 3 to 4 minutes to store the image and to send it to the cloud. So which was very much time, because if you are taking data of, let us say, 1,000 fruit sample on the field, and 3-4 minutes if your device is taking for every fruit, so you will be needing a huge amount of time. So on the bottom, whatever you are seeing, it is a recent version. In this we have used ready-made Raspberry Pi based computational platform, and based on around that we have this designed our readout circuit and this illumination independent algorithms and everything. Now this platform already has this BLE on the board and Wi-Fi on the board. So using these services, we have transferred data to smartphone based app and further to the cloud. And we have used two kind of illumination independent strategies. One is this: we have fixed a few of the LEDs around the camera to reduce the environmental illumination, and also we have implemented the software based correction to reduce the effect of illumination, environmental illumination. So this is the camera we have used, and this is the functional block diagram of the device. So these are a few of the field trials, just going for final, let us say, testing or final data collection and final training of the model. We have thought, let's test whether the device is working or not. So we've actually gone to the field and gone to the architecture, and we've seen that how much time our device is taking to store the image, and whether image is coming clearly or not, and how much time it is running, whether it is not taking too much power. These kind of experiments we have conducted. So we have just like I have told you that for hardware based illumination independent, so just like for software based illumination independent, we have taken images of fruit sample into different illumination, whatever LEDs we have fixed with the camera. So we have taken 7 different images for each fruit sample, and we have divided fruit sample into 2 different parts. So obviously using camera, you can only take the front part of the fruit sample, and your behind the fruit sample part will be remaining. So we have divided the fruit sample into two parts, and every fruit sample we have taken 14 images. 7 images for front of the fruit sample and 7 images from the bottom of the fruit sample. Then each fruit sample images has been taken into different illumination. And this W2 is in the blue, W4 in the can, W6 yellow. So these agenda was to taking these images into different illumination is to only implement software based illumination independent algorithm to compensate the effect of illumination coming from the environment. Then this segmentation based technique, different segmentation based technique have been implemented to actually extract the region of interest, because there are a lot of leaves on the fruits are there. There are a lot of other fruits also there. So we implemented this first technique, which is a contour based extraction, but it takes the performance of this method was not up to the mark, because it was working fine only when background is white and when limited fruit samples are there. So we have discarded it and implemented the second segmentation technique. So in this technique, we have used this HSV based color models and this Canny edge detection based contour extraction methods to segment the different fruit samples. And you can see that this method was working very fine, and even leaves are there, so there was no problem. So on fruit, if on tree there were multiple fruits, and it can segregate each fruit in your data. So these were some of the color features. So we implemented this k-mean clustering method to extract different colors present into the fruit sample. So these are the dominant color when one color present in the fruit sample. These are the dominant color features when two colors are present in the fruit sample. These are when three dominant color present in the fruit sample. And apart from these are some of the other features which we have used for training of the our models. One is this average color coordinates, then variance, then chromatic color coordinate, and obviously number of pixels. So this was till this we have developed this four different handheld device, and we have tried to figure out that what are the features. We have tested it basically. Now our aim was that how we can actually use this device into real scenario and for the real fruit samples. So for that we have to train all these devices for large number of fruit samples to generate the data set of our device, and also to generate the data set all of known parameters. That is, so basically we have generated data set of these 6 parameters which I have already mentioned: this weight, volume, chlorophyll, carotenoids, and total soluble solid, and sweetness and acidity. And based on that, we have tried to correlate with optimal harvesting time of the fruit sample. So these were the fruit samples collected from different nearby orchard, and we have labeled it. We have from sample ID S1 to S100, or let us say S1000. So this was the data generation protocol we have used. What we have done for similar fruit sample, we have taken data from our 4 different handheld device, and we have also conducted few of the standard experiments to generate the actual quality parameters of data set. So using our device, it is we have the sensor response data set, and using these methods we have the known data set of, let us say, what is the pH of the fruit sample, what is the chlorophyll of the fruit sample, what is the weight of the fruit sample, what is the shape, what is the size of the fruit sample. And after that, we have implemented different correlation model to correlate our response, our developed system data set with the data set generated using these methods. So this is the first method to generate the weight and volume. Obviously this weighing method has been used along with this Vernier calipers to measure the diameter of the fruit sample, and this is the generated data set of first 100 samples. And after that, we have extracted the juice of the fruit samples to perform these kind of standard chemical processes to generate the sugar data set, and also different glucose data set, and a total sugar data set. So apart from this, we have used this refractometer to generate the data set of total soluble solid. and also chlorophyll and carotenoid. So these are, whatever I am showing you, these are standard methods which are already known into literature, and we have used these methods to generate the known data set of the fruit sample. Then so after this we had this known data set of the fruit sample which we have generated using these methods, and we have the data set generated using our own systems. Then we can implement different correlation methods to correlate both of the data set. Then those models we have used to train our handheld device. So this was the chlorophyll data set generated of first handheld device. And this was the, so using this commercial imported pH meter we have used to generate the acidity data set. Then after this we have implemented this data analysis and modeling schemes. So before that, now we have our own system data set and standard data set. Now we have gone through two different statistical methods and a artificial neural network based methods. So basically in the statistical method, we have implemented three different techniques: multiple linear regression, and this partial least mean square regression, and this principal component regression, to correlate both of the data set. And in the ANN method, we have used these five different modeling schemes to actually correlate both of the data sets. So we have thought, so these methods we have implemented with different single sensing techniques. And what we have found that each single sensing techniques are not able to perform very well. So whatever they have shown the results, each for prediction of different quality parameters, like for chlorophyll, volume, shape, size, acidity, and all these parameters are pretty much low, even less than 70%. So there was no point to use those models for training our device. So because for each parameter accuracy should be minimum 90%. So we have thought to use these model, making no sense. So basically so after that we have gone to how we can combine the two different sensing techniques and use different data fusion methods to predict the information, because all four sensing techniques are complementary in nature, and each sensing technique has consists different nature of information. Like electronic nose consists smell information, spectroscopy consists internal compounds information, machine vision consists the information of outer surface, and this ultrasound consists the information of the stiffness and firmness. So if we can combine these information, might be there's a probability that our prediction accuracy got increased, and we can of the and testing into real time. So we have thought to implement different fusion of multiple sensing techniques. So we have actually combined different sensing techniques in all possible ways. So total 11. So there all three data fusion methods have been explored: this low level data fusion, mid level data fusion, and high level data fusion method. And in each combination we have tried to combine the data set, and we have seen what is the accuracy of each combination. So you can see in the low level data fusion, ENOS machine vision combination, ENOS spectroscopy combination, then obviously in the last all four combinations. So whatever combination we can make, and we can go for data processing and see what is the accuracy to predict these parameters. We have done that. So this was the architecture of the low level data fusion. We have combined all these sensing techniques and this sensing technique specific pre-processing methods we have implemented, which we have already used earlier time at the single sensing technique analysis time. Then we have combined them in using low level data fusion methods and extract the features, and implemented different statistical and error based modeling methods. So we have divided the data set into 70% and 30%. 70% data set we have used for training of the models, and 30% data set we have used for testing of the models. So we have after a lot of data analysis and iterations, I have not added those multiple graphs and multiple results in the presentation, because those were too much data. So I have added directly results what we have got from low level data fusion methods. You can see that for predicting the total soluble solid, the combination of machine vision, optical sensing technique, and electronic nose performs best using Bayesian regulation modeling technique, and using 6 number of neurons in the hidden layer, and the accuracy was 95%. So earlier the best accuracy what we have got using single sensing technique for TSS was around 70%. So it increased by 95%. Then this chlorophyll A for optical sensing technique with the combination of machine vision sensing technique with the Bayesian regulation modeling method with the new 8 number of neurons shows best accuracy around 95%. So you can see most of the accuracy, prediction accuracy of the quality parameter got increased and shown pretty much good results, except this volume and pH. So obviously this pH and volume we have not got good results, so we have thought to go for a next data fusion method, which is mid-level data fusion methods. So in the difference, what is the difference between low-level data fusion method and the mid-level data fusion method? The fashion to combine different sensing technique is different. Like in low-level data fusion method, we are combining different sensing technique after pre-processing methods, while as in mid-level data fusion method, we are combining different sensing technique data set after feature extraction. So basically mid-level data fusion is feature level fusion, and low-level data fusion is basically for your sensing level technique based fusion, or let us say pre-processing based. So in this also we have adapted similar modeling methods and similar fashion. So we have taken 70% data set for training and 30% data set for the testing of the our models. So after that, multiple iterations we have conducted using these models, and in using mid-level data fusion methods, we have found that this accuracy for different parameters, for some parameter it got decreased compared to low-level data fusion, and for some parameters it got increased. But again, this data fusion practices perform far better compared to single sensing technique results. You can see that most of the accuracy is quite greater than 90%. Then after that, this high-level data fusion method is implemented. This mid-level data fusion and low-level data fusion generally have been used to predict the quantitative value of the fruit sample, quantitative value of the any parameter, like what is the value of chlorophyll and what is the value of, let us say, acidity. While as for high-level data fusion is to predict the decision of the fruit, decision of any kind of case. So that's why we have called it identity declaration, identity-based fusion. So in this, basically, we have declared 3 identities, which we have already declared, like maturity and ripeness and flavor. And for each identity, we have thought which of the this parameter shows best results. So this these sensing technique, along with for these quality parameters, have shown best results. And so finally, we have combined this high-level data fusion methods using different sensing techniques to predict the quality of the fruit samples, like ripeness, maturity, and flavor. So ripeness has been correlated with the chlorophyll A and chlorophyll B. Maturity has been correlated with the weight and volume, and flavor has been correlated with the sugar, total soluble solid, and acidity. But you can see the accuracy was not quite good. Still, the accuracy was 89% for chlorophyll A and 89% for chlorophyll B, and weight volume also very less. So the overall decision accuracy was also not very good. So till now we have developed model for 3 different data fusion practices, like low data fusion, mid-level data fusion, and high-level data fusion. So we have thought how we can combine all these 3 models and come up to single model, like for high-level data fusion combined with the mid-level data fusion and combined with the low-level data fusion to extract the best possible model to predict these parameters. So these are the ranges decided based on multiple experiments for unripe, partial ripe, total ripe, and for maturity also, and for flavor also. And this is the decision tree algorithm which we have used to correlate the ripeness, maturity, flavor with the harvesting decision. So we have designed the 3-layer based decision tree model. On the first layer, we have tested the maturity using the weight and volume multiplication data. On the second layer, we have found calculated the ripeness index based on 3 different classes, and the last layer we have calculated the flavor index assessment using TSS and acidity data. So in this model, whatever sample citrus sample came in, it basically first gone through maturity screening, and if sample is totally mature, then it passed to second layer of the model, otherwise it got rejects. And if a totally mature sample passed to a second layer of the model, and if that sample is also totally ripe, then it only passed to final layer of the model, otherwise it got rejects. And if a totally ripe sample with a totally mature sample have best flavor index, then only it suggests that your sample is ready to harvest. And if it is having average or poor flavor index, then it not suggest that sample is ready for harvest. So this was the final results by combining all the data fusion results from low level data fusion to mid level data fusion to high level data fusion. So this for TSS, low level data fusion results have shown best to combine the e-vision, optical and electronic nose, and using this Bayesian regulation technique, and this accuracy was around 95%. The chlorophyll for also above 95%. The pH were around 78%. So except volume for each parameter was quite good, and so these parameters we have converted into final model which we have used. You can see this is the final model which we have used to train our device, which have a final single device. So to predict the chlorophyll A, we have combined all 4 sensing techniques with these kind of baseline correction methods, and this scaling and normalization methods, and this feature extraction methods. And after that, this for chlorophyll B also, this combined all 4 methods using this application specific pre-processing methods and feature extraction methods and correlation methods. And for this, let us say this acidity also, and total soluble solid, and weight and volume. So all this further has been combined to identity level fusion, which is the total chlorophyll and flavor index and maturity index. So ripeness index has been calculated using chlorophyll A and chlorophyll B, summation of chlorophyll A and summation of chlorophyll B, because ripeness is inversely proportional to chlorophyll. And flavor has been correlated with that division of total soluble solid and with the acidity, because total soluble solid indicates the sweetness of the fruit sample, and acidity indicates the sourness of the fruit sample. So the ratio of this index will tell you actually the how much your fruit is sweet and how much less your fruit is sour. So based on that, it is the flavor index we have predicted, and the maturity index we have correlated with the multiplication of weight and volume. And all these decisions we have combined to predict the harvesting decision, whether fruit is ready to harvest or not, using a decision tree algorithm, which I've already shown here. This is the decision tree architecture which we have used. So this is the field trials and tested. So after developing all the instruments, we have gone through all around the India, different orchards, even ICR Nagpur. They have this 1 acre of orchard, and they produce 9 different varieties of the citrus. So we have thought to go there, because all around the India only 7 citrus varieties are found, but they produce 2 more hybrid varieties of the fruit sample. So we have tested our devices after training of models for all 9 varieties, and the results were quite fine. And these were also this demonstration in different festivals with the ICR, CCR lights. So you can see the size of the fruit sample in the top of the image. So this is the pomelo, which is one of the breed of the citrus sample. So our device, few of our device also got published in CSIR news also, and we have demonstrated into different festivals. In the right hand side, you are seeing the Bhatkar sir, who actually developed our first supercomputer in India. So we have demonstrated them, and they have quite appreciated the device. So this was the references of the my talk. Now, if you want to ask anything, I am open for questions.
Yeah, I request all the participants, if you have any queries, you can raise your hands. Or you can put your questions in chat or in a WhatsApp group so that I can ask your queries to the our today's expert. Yeah, Mr. Dr. Anup Vibhuti is there. Sir, I'm allowing you to unmute yourself. Sir, please. Dr. Anup Vibhuti, sir, please unmute yourself. Dr. Anup Vibhuti.
Maybe he can write in the chat box if his mute.
Yeah, please write your query in chat box.
He write that and he's unable to mute, so you can also unmute him.
Actually, I am not able to unmute him, but I have the control to give them access whether they can unmute or not.
Okay, he can write his question. Yeah. Yes, Dr. Gayatri Padeji. Obviously, so basically, number of neurons in the hidden layer does not has the actual relation with the parameters, but the number of neurons in the hidden layer basically signifies you that if you are handling high complex model, that you need to use the high number of neurons, and if you are using a, let us say, less complex model, which is having a less non-linearity between target models and input models, then you can then you can use less number of neurons, because basically the aim to use these kind of models is that if you are having high amount of non-linearity between your target parameters and your input parameters. So if you are having high level of non-linearity, then you need to increase the number of neurons in hidden layer. And if you have, let us say, less amount of non-linearity between input and output, then you can use less number of neurons. And this whole process to optimize the number of neurons is basically hit and trial method. You need to change the number of neurons and then again see the prediction accuracy, and also prediction accuracy along with mean square error. And whenever this mean square error got stopped decreasing, when you have, let us say, when you receive the minimum number of mean square error, then you can stop changing the number of neurons. Okay, just a second. Second question is, I think. Yeah. Second question from Dr. Anup is this: is there an facility to have MOU? Yes, Dr. Anup, definitely we can have MOU, and we need to found a common interest between your institute and our institute, and we need to discuss the agenda, and we can have definitely MOU for joint collaboration. That possibility is always there. Sir, whether
Yes, because my PhD itself is in this area only.
okay.
I have done my PhD on the basis of nutrient and pesticide management using image processing.
Okay.
For grapes, I have did it,
Okay.
and I have one patent published also, but
Okay.
it is in final process. So presently I'm working on various parameters related to soil and fruit volume and management, like parameters also.
Okay.
So that's why I'm asking whether is it possible to have a MOU with CEERI.
Definitely, sir. I already told you that what we can do that after this talk. We have to find a common schedule in which you are also there, and I am also available, then we can fix a meeting on this Google Meet or whatever this Microsoft team, and we can discuss the common agenda, and based on that we can initiate the MOU. That is not the problem.
Okay, thank you, sir.
Okay. So next question is from Pranali. Sir, whether the device is in the market for checking the attribute of the... Yes, Pranali. So we have developed the final device in which all four sensing techniques are there, and using this final model, which we have got after a lot of iterations, and this technology transfer process is there. We are going to transfer this technology to a company. There are 2 to 3 companies are there, so they'll take this technology and then they'll go for mass production, and then this technology come to the market. So it will hardly take for next 6 months to come to the market. While as right now we have already given few of the products to the different Indian fruit growers to get their feedback. Then this Nitin is there. Is training and testing percentage fixed and certain or okay? So Nitin, this is very standard kind of protocol with any kind of machine learning or deep learning model algorithms. Generally, this is the suggested that 70% kind of the data set you need to take for training and 30% for testing. While as it can be changed definitely, but the training quotient should be more. It is not like that training quotient should be 30% and testing will be 70%. Yes, Radhika. So you have asked that can chlorophyll content can be monitored from remote place. So whatever device, Radhika, we have developed, it is a handheld device. So a fruit grower can just touch, even not touch, just take this device close to the fruit sample, and it will predict all possible parameters along with chlorophyll, total soluble solid, and this weight, volume, acidity, and overall maturity, overall ripeness, flavor index. And this data goes to cloud, and anyone around the country or even outside the country can access a website which we have made to see the overall yield of that fruit sample, or let's say history of that device, and he can see what is the testing history and what is the parameters and everything, along with the location information and date time stamp. Manoj asked that the device is applicable to all the fruits. Manoj, actually these kind of right now whatever device we have developed, it is trained for this citric kind of a fruit samples like lemon and citrus and different breeds of citrus. While as right now we are in the process to generalize this study, because if we want to generalize this study, we need to do this kind of iterations for all fruit samples, and we are right now in the process to generalize this fruit a device for all fruit sample. Right now this is only applicable for citric fruit samples. Any else question?
I request all the participants, if you have any query, either they can write in a chat or now
Yeah,
they can.
Yeah, I have a question, Praveen. Manoj asked that, but in case of grapes, will it work? Manoj, that is the beauty of our overall research. We have not used single sensing technique, which I have explained in the earlier when I was showing the device available globally. So our device have 4 different sensing techniques. So that's why it can be generalized for different fruit samples, because grapes is also having this acidity quotient and this chlorophyll quotient and this sugar index quotient. So this spectroscopy part of our device and electronic nose part of our device can definitely predict the quality of the grapes fruit samples. So that's why this idea came into our mind, because if we want to develop a device which can be work for all fruit sample, we cannot go for one sensing technique. We need to use multiple sensing techniques. That's why we have conducted these experiments, and we have also found that one sensing technique is not enough. So one sensing technique, results of one sensing technique, single sensing technique, I have not shown to you guys. But I have results with me. Results for each sensing technique to predict these parameters are quite low. So if you talk about any of the sensing technique, results were less than 70% for any of the parameter. But when we have started combining two or more than two sensing technique, results got increased and results got very much good. So that is the basic idea behind it, to use multiple sensing techniques, and it can be used for a series of fruit samples.
Thank you, sir. Some of the questions I have received through WhatsApp.
Yes,
How
you can ask.
how you have selected the hardware device for your IoT application?
Okay, so the idea was behind before selecting any kind of hardware or computationally, let us say, platform, we need to select the application, because it is very much dependent on application. So our application was that whatever device we are going to develop, it will be using in the field. So there are 2 to 3 things we need to keep in mind. That first is the power consumption, that then second is the network issues, then third is the portability of the device. So basically while selecting the platforms, we have thought for all these things, and we have not, in whatever device I have shown you, we have not used any kind of ready-made platforms, excepted in our machine vision sensing technique. We have used ready-made Raspberry Pi computational platform. Except that, we have used our indigenous hardwares, we have used our indigenous IoT enabled solutions, we have developed our own indigenous IoT enabled services to enable our device for sending data from device to mobile and then mobile to cloud. So we have not used these Arduino boards and these kind of things. We have developed our own boards and our own computational platforms, and we have thought of that how it can be consumed very much low. So whatever device I have shown you, except that camera based device, it takes very much low power. So if you talk about the data, so it takes around roughly 100 microampere standby current and 10 milliamps operational current. So you can see the small lithium ion battery with 1000 milliampere per hour can run these devices. Still, whatever battery we have used for our device, that is 1000 milliampere per hour. It can run around 2 to 3 days without charging. How chlorophyll is detected? So basically, Gayatri asked that how chlorophyll is detected. So basically, Gayatri, we have not detected chlorophyll. We have predicted chlorophyll. So what we have done, we have collected around 1000 fruit samples, and we have first generated the chlorophyll data of that those fruit samples. So what we have done, we have implemented this Enthron method. Just a second, I will give you the exact method we have used, chemical method we have used to generate the chlorophyll data set. Just a second. Yes, so this the Enthron reagent based method, it is well explained in the literature. So what in this method, they actually extract the fruit juice of the fruit sample, and they use this Enthron reagent into the fruit sample, in the juice of the fruit sample, and that juice sample change the color, and that change in the color we have tested with the portable spectrometer and generate the data set of the chlorophyll. So this is the process we have adapted. Then after this generated of the chlorophyll fruit sample, we have correlated with our device data set. Then we have found the pretty much good accuracy. Then we have our device is predicting this chlorophyll and all other parameters without using that chemical method. I think if it is understood, please let me know.
Thank you, sir. There is another question, that whether the same procedure that you did for the citrate type of fruits will be applicable to other fruits also, the approach?
Yes, a similar approach will be. That's why before even initiating this idea, we have found that what are the sensing techniques, what are the combinations, what are the data fusion methods we need to use, which we can came up to final device which can be useful for all kind of fruit samples. So right now we are trying to generalize for mango, because mango is one of the problem in India, and there are huge chunk of amount India export every year to US, UK, and this obviously this during this Congress government, you guys are aware this Alfonso issue. It was a big fame for India. So we are thought to develop this handheld instrument for generalizing this for first mango, and then we will try to generalize for most of the fruit sample which are very much available in India.
Uh, so thank you, sir. I think from the participant there is no query, uh, so we'll stop here.
Okay.
So, yeah, thank you, sir, for delivering a talk on this non-destructive sensing techniques for the citrus fruits.
Okay.
I hope the approach that you have used for the development of these products will help the participants to do more with their IoT applications. So we'll stop here. Sir, would you like to continue with the hands-on session right now, or would you like to take some break?
It depends on the participants what they want. I have no issue.
Actually, with the schedule, we have a break of 15 minutes. Is there?
So definitely we can break it, no issues.
Okay, sir. So we'll take a break of 15 minutes, and at 4:30 we'll start with our
Okay.
next session. Yeah.
Okay.
Thank you, sir.
Okay.